morning, good afternoon, good evening, wherever you're located. Thank you for joining us today. As is typical for these webinars, I'm going to try to stall for a minute or two. I wanted to send some positive thoughts and offer your own thoughts and prayers for Kate Mitchell today. She's in the hospital. What sounds like getting some fairly routine treatment, but at the same time, just wishing her a quick recovery. We'll get into the details of this agenda in just a moment. I'm going to give it one more minute till maybe 10:03. The participant numbers are still going up. I'll start off by saying that if you have an interest in the seeing closed captions, there's a live transcript button on the bottom right side of your screen. It says live transcript. If you want to activate it, you can hit live transcript and then hit show subtitles to be able to see the subtitles on your screen. Okay, I think we should get started. Kate, do you mind going to the next slide? So again, these webinars, can you go back to the first slide? Thanks again. So these webinars are built specifically to help inform you of current actions of the Mammalian community, and specifically the work of a foundation building strength towards finding treatments for Mammalian myopathy. Your input is critical. I'll go over a few bullet points in the first two webinars of things that we've learned in a minute already, and the sharings that we're starting to initiate. The steps that we take are meant to be inclusive to everyone with NM. So please, if we happen to do or say something that feels like it's not inclusive, please reach out to us and let us know. And we're open and want to have full humility in the way we treat everyone with Mammalian myopathy and the even like diseases around us. We're trying to support not only Mammalian myopathy, but everyone that we can in our circle. All right, well, let's move on to the next slide. As we indicated, captions are available for these webinars now. The live transcript button on the bottom right of your screen, feel free to click that. Though that will also produce a transcript of the webinars. Obviously, sometimes those are jumbled and not interpreted 100% well. So we will try to clean up the transcript. Recordings will also be available for every webinar. So please watch for those communications. A few things that we've learned already, that tissue deposits actually can be done across your lifespan, which actually can be quite helpful. So not only at that first muscle biopsy, but as you've had any further procedures or surgeries, it is helpful to actually have some future deposits done, so we can see the transition and what's happening to your muscle over the course of the lifespan. That is also true for blood draws. Any blood draw you're having, you are capable of sending those cells for cell line development. And so those are both handled by Stacy Cossette on our team. And they handle basically everything, every step of the way. They'll get you the kit that you need. They will communicate with your medical center and handle those transitions. I just wanted to indicate there were some questions about translations for our care guide. I believe that it's in 4 different languages in its current state. It's been translated certainly in English, Spanish, and I believe it's in Russian and Polish currently. We're looking for other families that would be willing to help us translate the guide into future other languages as well. Okay, we can go to the next slide. Right, so reviewing again, the primary goals of these webinars is to actually move translational medicine forward. And you're going to hear a lot about translational medicine today. Review our timeline for the objective impact that we hope to have over the next 5 years. 5 years sometimes seems like a long ways away, but we've got some steps to take. And again, I think you'll learn a lot more about those steps today. A little bit deeper dive on the impact that our patient community can have. So you're going to learn a little bit more about action steps that can be taken. And then review our current research, the 5 or 6 projects that we're funding currently. Once again, this is our team. I want to be super thankful to them. I will say that everyone on this team has been super loyal, attempting to be very inclusive, and their intent is to do only good. I can say that, and working very, very hard, hours and hours and hours a week to help pull together not only these informational webinars, but all of the work on the back end to help move research for Mammalian myopathy forward. Okay, the timeline, as we've reviewed, over the next 5 years, really we hope to be launching into clinical trial. So working backwards from that, we need to understand the effects that CRISPR can have on NM animal models. We need to understand what medicines cause zebrafish to be more active, and we're close to that. We hope to increase the CMDAR, the Congenital Muscle Disease International Registry participation by 100%. We hope to double our numbers by the end of the year. And I'm happy to say we've taken a step forward for that. We've actually confirmed hiring Sam Jacobson. She's going to actually work for us part-time to actually improve our registry numbers. Great to have her on board. Now, by next year, we hope that 2 companies in industry actually have NM tissues in their labs, and they're testing it with their own small molecules and different compounds that they've developed. In 2024, I will say 2025 at the latest, we hope to have a fully complete natural history study. You're going to learn more about that today as well, and a better understanding of the pathophysiology of the rarest cases of NM, not only those that are the big numbers like actin and nebulin. In 2025, we hope to identify the best NM treatments to be able to start preclinical studies and establish an interaction with the FDA with agreed on protocols and pathways to regulatory approval. And then 2026, again, starting to launch that clinical trial. And we're going to learn what the language of clinical trial actually means in a bit here too. And then again, finalize the discovery on genetic repair for a curative mechanism. All right, a few terms. You're going to hear a lot of different terms today. These are a few that are going to have some repeating input. So the NCATS. NCATS is the National Center for Advancing Translational Sciences. So it's government funded. It is an institution that we're going to ask each of you to, at some point, dig into that website, which we have the link for you today, and read a little bit more detail about how you as a patient or family partner can actually participate and actually support the cause of translational medicine. NIH, which is the National Institutes of Health, founded many years ago, and it's actually government funded to actually support rare disease development discovery. The FDA, Food and Drug Administration, EMA, the European Medicines Agency. Okay, here are a few words that are important to understand for today. The natural history study, that is a standardized collection of information about the affected population to record what disease course entails when no intervention is performed. So we want to understand the path that someone's life with Mammalian myopathy goes through, whether they have, again, a more rare form of the genetic variant, or they've got one of the more common ones. It's really important when we're going to execute an outcome measure or a clinical trial that we actually understand what no intervention does first over the course of their lifespan. So that's why engaging in the natural history work over the course of several years will help us watch and look at the behavior change and the phenotype change in the population that we're serving. An outcome measure is a standardized method used within the medical practice or research, which is used to assess the effect, in both positive and negative, of an intervention or treatment, i.e. Grip strength, functional vital capacities or breathing strength, walk test, or even a slurp test for bulbar function. So those are outcome measures. A clinical trial is, again, engaging in a research study in which human subjects are prospectively assigned to one or more interventions, which may include a placebo or other control, to evaluate the effects of those interventions on high-related biomedical and behavioral outcomes. Now, we're actually imparting some trials that you'll hear about today on animal studies, so you'll hear a little bit about those in a bit here. So I think the next slide is going to allow me to introduce you to Gus. Gus is our scientific director. Hang on for a second. Yeah, here you go, Gus. I'll let you take over.
Hello, everyone. In our previous webinar, we talked about the importance of research resources and how you, the affected families, can participate by registering in the CMDIR or other registries as the one in the BEX lab, and donating cells and tissue for research. And today we will continue discussing our role as the Mammalian myopathy affected community in helping the research move forward. Moving knowledge from bench to bedside is called translational research. Next. So translational research, the goal of the translational research is to move the basic science discoveries more quickly and efficiently into practice. Translational research seeks to produce more meaningful and applicable results that directly benefit human health. What does translational research do? Encourages and promotes multidisciplinary collaboration among laboratory and clinical researchers. And we, as a patient organization, we do our part with events like our scientific and family conferences by facilitating the exchange of ideas and data to stimulate collaboration among the different stakeholders, researchers, clinicians, and the affected community. We also get involved in this process with our grants program, which funds different research strategies and also stimulates collaboration between our grantees. Next. Translational research also incorporates the voice of the patients, their needs and opinions for treatment options and care. Translational research is changing the concept that patients should be partners, not just research subjects. And this PICORI project that we are doing with this series of webinars and other activities that we will be doing seeks to help in this step of the process by bringing closer researchers and the affected community. Another fundamental tool here is, again, our registry, the CMDIR. The information you provide and the surveys that you complete capture valuable data for translational research. And again, our scientific and family conference, which facilitates direct interactions between you and the research community. Next. Translational research also helps identify and support the adoption of best medical and health practices. On this side of the translational research, AFBS has been key in developing the standard of care guidelines for congenital myopathies. You can find the guide in this link here on our website, also at the CMDIR and the tissue repository websites. And also I learned yesterday from Stacy that there is a Kindle version that you can get in Amazon. Next. So now let's watch a short video about translational science done by NCATS. As Mark explained, NCATS is the National Center for Advancement of Translational Sciences. It is the newest center, is the newest center created by the NIH. It was established in 2011. So please, Kate, play the video.
Every day, scientists around the globe make important discoveries and breakthroughs in understanding illness. Turning these discoveries into improvements in human health is difficult.
Is it
The
working?
drug development journey costs too much, takes too
Yes.
long, and fails far too often. Meanwhile, thousands of diseases affect human health, but treatments are only available for about 500. This represents a challenge, but also a huge opportunity. There is tremendous need for people to discover, develop, and disseminate the next generation of science and technology to improve human health. This process is called translation. Translation is the multi-step process of turning observations in the lab, clinic, and community into interventions that improve people's lives in the form of diagnostics, therapeutics, medical procedures, devices, and behavioral changes. The field of translational science requires people with diverse skills, including biology, chemistry, computer science, engineering, medicine, and public health, to come together and work as a team in order to make a real impact. The fundamental characteristics needed go beyond individual specialization. It includes a broad understanding across the translational spectrum and being a team player, a rigorous researcher, a systems thinker, a skilled communicator, a domain expert, a process innovator, and a boundary crosser. These translational scientists are explorers working in teams, innovatively and collaboratively, searching for ways to break down barriers and improve patients' lives. Recently, translational projects have developed new medical devices, created gene therapies for rare diseases, built tools that support patients in their engagement in the drug development process, using resources to advance biomedical data sharing, repurposed existing medicines to treat other diseases, and advanced drug development processes. The field is intrinsically rewarding because of the impact one can have on the science of developing new medicines and improving the lives of patients. Translational science presents tremendous opportunity for innovation and systematic change, developing solutions that make the process of developing a new medicine better, faster, and more efficient. Translational science is the youngest science with boundless promise to transform biomedical research and medicine. It requires innovative thinkers with a patient-focused approach to ensure that the patient's perspective is included in every step of the research process, breaking down barriers and collaborating to prevent, treat, and cure disease. If you want to make a real impact on the lives of millions, learn more about the field, or become a translational scientist, please visit us online. Join us.
Okay. Next, as we heard in that video at the end, they emphasize the need to have a patient-focused approach to ensure that the patient's perspective is included in every step of the research process and trying to break down barriers and collaborating to prevent, treat, and cure disease. This is now recognized not only by the NIH and NCATS, but also by pharma industry and regulators, in the FDA, the EMA, and other regulatory bodies in other countries. And that's why, I mean, your voice now supports rare disease treatment development. And NCATS has a toolkit. They put together a toolkit with reliable information about the stages of the therapy development, the processes, and the ways we can become involved. And the ultimate goal is to help us become a driving force behind the development of therapies. So we would recommend the affected community to read this toolkit for patient-focused therapy development. The link is there. And you can submit questions, opinions, ideas about the toolkit and the research process in general, and for Mammalian myopathy in particular. Going at your pace in this guide would be good for you to prepare for better discussions and understand better the next series of webinars that we will be doing. And this talk focuses on the translational research. So here is a screenshot with different tabs of that toolkit. The one open that you see is the glossary tab, which will help you understand familiar terms. And indicated in the red circle are the tabs most important to the Mammalian myopathy community at this point in time, as we are in this stage of trying to translate discoveries to be tested in humans. Then the other tabs are more related already for when clinical trials are already going on. Next. I hope this is helpful to wrap up this part. Here is a flow chart showing us the translation from basic science discoveries like disease mechanisms and targets for potential therapy interventions to human studies, where those interventions are tested for efficacy. And ultimately, translation of that data into clinical practice and health decision making. Next. And the translation research in mammalian myopathy, we have been doing a good job on the base of the translational research flow, the left side of the figure, defining mechanisms, targets, and potential therapeutic modalities. Now, we also need to activate the translation to testing potential therapies in patients. And all the discussions about treatment developments end up telling us that natural history studies is fundamental to select the adequate outcome measures for future clinical trials. And a big factor of why too many treatments fail in the clinical trial enterprise is because inadequate knowledge of the stages of the disease. How is the progression, the variability, etc. We are with a group of international experts. We have been working with in planning a natural history study. We actually have a drafted protocol ready. And these studies are very expensive and need a lot of coordination. And we are looking for ways to finance the costs and put together the multiple site team to conduct this study. So we will be talking about this in future meetings. And again, affected community participation is key. Ways that you will help is by updating your CNDIR profile and answering the surveys there that give us a lot of data about your preferences on the procedures, the protocol developments, and then you will the study will depend on your willingness to enroll. And so another good opportunity would be to join Family Advisory Board meeting. The first one, we will have one during our Scientific and Family Conference on July 1st. And again, thank you, and again, encouraging you to continue donating, continue supporting the CND tissue repository and our cell bank by donation of your cells and tissue. So meanwhile, while we prepare for the natural history study, let me give you an overview of the current funded research in the preclinical stage that is forcing the translation steps towards clinical trials. Next. So let me remind you that the congenital mammalian myopathy are a rare genetic disorder. There is a calculated incidence of 1 in 50,000. That's based on one study in Finland. So if these numbers are correct or approximate, we should be expecting that in the US we have about 7,000 cases, and worldwide about 150,000. And the congenital Mammalian myopathy is caused by 12 genes that we know today, where Nebulin and ACTA1 are the most frequent ones, and we have other 10 that are less frequent, but not less important. And probably there is now a 13 gene, ABS-SL1, that also seems to cause some forms of Mammalian myopathy. And as you know, unfortunately at present, there are no proven disease-modifying therapies for Mammalian myopathy. And we are stimulating the knowledge, the generation of knowledge with grant programs. And at the moment we have 6 active research grants. And I will talk a little bit about all of them now. Before reviewing each funded project, let me remind you where in the skeletal muscle is the origin of the problem in Mammalian myopathy. As you will see in the next slides, here the figure shows that from the intact muscle, going deep in the muscle, in the tissue, to see where the problem is. If you go to the sarcomere, that is the smallest contractile unit in muscle, is formed by thin and thick filaments. The myosin is forming the thick filament, and actin is forming the thin filament. And the thin filament, all the Mammalian myopathy causing genes encode proteins that are either components of the thin filament or contribute to stability or turnover of thin filament proteins. And so it is at this level of the contractile unit where the thin and thick filaments interact, where muscle weakness originates in Mammalian myopathy. And as you will see in the next slides about each individual project we are currently funding, I want you to notice that we tackle multiple strategies in parallel. So let's see, one of the projects is called a drug proposing for the treatment of Nebulin Mammalian myopathy, led by Robert Bryson Richardson at Monash University in Australia. And he's following a previously funded project that tested a total of 1,400 FDA-approved drugs on Mammalian myopathy affected fish with Nebulin mutations. In the picture, you see the normal fish and the fish without Nebulin, and in the pictures with the green fluorescence, you can see that the very first picture shows the severe phenotype of this fish with the muscles that are a little bit disarranged, and it shows some Mammalian bodies too. Next. So they are now doing a deep testing of the 5 test candidate drugs. They selected at the beginning about 20 that did some effect, but then they work with those, and now they find 5 candidates that have a strong effect on the fish muscle, and they will be doing those response scores for long-term survival and measure muscle functionality. Next, they will test 2 treatment regimes, beginning from giving the drugs at the beginning from birth, with the idea to see if the drug can prevent or delay the loss of function, or commencing the treatment after the onset of disease symptoms to see if the function can be restored after the symptoms started. So this is the idea of that project. The next project is correcting muscle function in mammalian myopathy by mutation-independent approaches. Is led by Vandana Gupta, the Brigham and Women's Hospital in Harvard. And she found also in a previous grant that mammalian myopathy muscles show abnormal accumulation of a protein called NRAP. NRAP stands for nebulin-related anchoring protein. This is a chaperone protein that helps the function of nebulin. And this abnormal accumulation contributes to disease pathology. So the experimental, the aim one is try to downregulate the NRAP and see if that result in the restoration of skeletal muscle deficit observed in mammalian myopathy. They are using the zebrafish model, mutant for nebulin. In the model, there is this increase in NRAP, and they are trying to reduce that increase by different approaches using small molecules or genetic tools. And so if they can normalize, the idea is to see if they can normalize the NRAP and improve pathology and function. And the second aim is very different to the first one. This is an approach where they are using a knockout mouse. It's a mouse that doesn't have one of the other genes, no nebulin, no ACTA1, is the KLHL40, that the mouse has this strong phenotype, is small, survives less, and is weak. And they will try to do gene correction by adding the KLHL40 with an AAV vector. So with a viral vector, they will inject the gene, and with time, they will try to they will evaluate if the addition now of the good gene can restore, can revert the disease phenotype of these mice. Other of our grants is to Henk Brancière at University of Arizona. He's studying the effect of myostatin inhibition on muscle size and muscle function in a novel mouse model of typical mammalian myopathy. Myostatin is a protein that prevents muscle overgrowth. In the picture, you can see a normal mouse and a big mouse that is a knockout for this myostatin. So when this mouse doesn't have the myostatin, so they have a muscle overgrowth. And in preliminary results, the myostatin inhibitor showed increased body weight and muscle size by about 20% in this new mouse model. And the one of the aims of this project will assess the effect of the myostatin inhibition on muscle mechanisms, force, size, and strength. So the question is myostatin inhibition a possible therapy for mammalian myopathy? That's what the researchers are trying to find. And they have a second aim that is also a gene therapy approach in two different nebulin mouse models. And they will do here, they will intend that overexpression of KLHL41, another form of another gene that causes mammalian myopathy. And this KLHL41 is nebulin chaperone protein, and the team anticipates a reduced loss of nebulin and preservation of muscle functionality with this treatment. Another interesting project we are funding is testing novel genetic therapies for ACTA1 mammalian myopathy, harnessing patient cells. Here is a clear evidence of what we were talking in the past webinar, and now that the importance of your participation when you are donating cells. In this case, the researchers have a few lines of cells from ACTA1 patients taken from blood, and they culture in a dish, and they have a technique to reprogram those cells. So they induce pluripotency stem cells. So now these pluripotent stem cells with different factors, you can grow in different tissue, like muscles or cardiac muscle. And that serves as a platform for testing precision medicines. The original proposal is to use CRISPR-Cas9 to knock out dominant ACTA1 mutations without harming the normal copy. So the wrong copy will be, that they will try to knock it down and only express the protein produced by the good copy of the gene. And another approach is a little different, that is the idea of gene replacement in both dominant and recessive ACTA1. They are using experimental genetic tools to provide the cell with instructions for turning on an alternative version of the skeletal muscle acting gene, the heart acting gene. The idea is to provide the cell with a similar protein that can perform the same function as the defective protein. And also, sorry, go back to the previous one. And talking about collaboration between researchers, this group is now in contact with another researcher that approached us for funding, that he is also working in a similar fashion, like using this type of protocol, producing stem cells out of patient cells. And they also developed a technique to generate also 3D muscles. So it's even a better model, because now you have a 3D muscle that you can measure other things like force and things like that when you introduce potential treatment. And they are working collaboratively now, and we have some plans for the future and more funding for them. Next, oh yeah, this one. So I think this is the previous to the last project that is looking at the disease from a different point of view, because they are trying to uncover the mechanism of myosin dysfunction in mammalian myopathy as a potential target for therapy. Remember I mentioned, here in the picture, you can see that the sarcomere has two very important filaments. One is myosin and the other is actin. And so despite the difference in the causative gene, the myosin binding to actin filaments is often disrupted. And the main function of myosin molecules in skeletal muscle is to produce force and motion. So we have there are a few pharmaceutical companies that are now targeting myosin to counterbalance muscle dysfunction in a number of disorders. And this study aims to uncover how myosin is dysregulated in mammalian myopathy, and they will be testing some of these compounds from pharmaceutical companies. And achieving this would provide a better ground for developing efficient therapeutic interventions. Sorry, I didn't mention this. Research is done by Katerina Pilin and Shini Laitila in Finland, and now collaborating with Julien Ochala in Denmark. Next, this is the last one. This is a grant that was not in the regular cycle. It was asked after the regular cycle with some money donations that we obtained later on, and is continuing the work of preclinical testing of a CRISPR-Cas9-based therapy for mammalian myopathy by Dr. Jim Dowling at SickKids Children's Hospital in Toronto, Canada. In the previous granted project, the group of Dr. Dowling explored for the first time the potential application of the Nobel Prize-winning CRISPR-Cas9 gene editing technology to mammalian myopathy therapeutics. They successfully created the specific guides and templates to edit and correct the most frequent recessive mutations in mammalian myopathy, that is the deletion of the exon 55 of the nebulin gene. They tested these tools on isolated cells with encouraging results. And now with the new funding, the research team will work on a critical step for the clinical translation of this novel strategy. They will now test these specific CRISPR-Cas9 tools that they created in vivo in exon 55 deletion carrier mice and exon 55 deletion patient-derived myotubes. If successful, this project will provide the proof of concept required for the advancement of this strategy to the clinics. And the next slide shows in a graph, in a graphical way, again, the most common net mutation is the deletion of exon 55. It means that part of the nebulin gene is missing. And using CRISPR-Cas9, they will add the CRISPR-Cas9 tools and the targeting template to repair the absence of exon 55, again using viral vectors, and they will do injections in the mutant mice and analyze and assess the improvement in survival, in muscle structure and function. And they also will continue in the bottom part that is on patient-derived myotubes with the exon 55 deletion. In the first picture, you can see this, these myotubes staying with different fluorescent proteins that stain actinin in red, the nucleus of the cell in blue, and the nebulin in green. And you can see that there is no nebulin because these patients have the exon 55 deletion. But when they reintroduce with the CRISPR-Cas9 the exon 55, you start to see yellow, that is the mixing of the red and the green. So now nebulin and actinin are expressed, and it seems that they are interacting. They have to optimize this process because the efficiency is not that good yet. And they will be doing that during the course of the grant cycle. Next, this was a brief review of the original projects. You will hear updates on these projects and interact directly with the researchers in our upcoming Science and Family Conference. So please register. There is the website for registration and information about the CMD, the Science Fam meeting. And we will have also these researchers in future webinars and meetings to interact with you and learn from each other. Because they, as we mentioned for this PCORI project, the idea is to have a conversation and you understand the research and the researchers understand your needs, your desires, your opinions. So yeah, next, before wrapping up, just to mention that we have a regular call for grants, which is every 2 years, but we are also available year-round to researchers and clinicians to get in touch with us about submitting proposals for evaluation by our scientific board. And our research grants are open internationally, so you can spread the word wherever you are. So we are planning the next, oh, go ahead, Sarah.
Gus and Mark, thank you for that fantastic presentation. And I think what Gus was leading to is that we have more webinars like this planned in the future. And our current placeholder for the next one is going to be on September 17th, 2022. So you can look for more messaging about that coming soon. Next slide, Kate. Thank you. And just now, Patty wasn't able to make it with us today, but this would be her plug for talking to everybody about what are some actions, because a lot of people are like, okay, this is all great. What's the next steps? And so here's a couple things you can do. You can take a look at the toolkit that Gus talked about in this talk. And that's for a deeper dive to look into this toolkit that's available. And the link is in the chat. Thank you, Kate. And also reach out with any questions. Make sure to stay connected with us if you have any questions about that. And I really, really hope to see you either in person or online at SciFam, and you can register for that. So, and if you have any questions, take a look at the conference format. It's just going to be a continuation and expansion of this project and some of the, interacting with clinicians, researchers, and each other. So I really hope to see you there. And then also we would really definitely want to hear your voice. And so we have a post-webinar survey, and we definitely got very interesting and good input from the last post-webinar survey and trying to incorporate some of that feedback into this webinar. So please keep it up, and you can look for the link in the chat, and we'll also be sharing it on social media. So thank you so much for all the speakers. And I'm going to turn it over to questions. And I'm also going to mention that one of our researcher scientific partners is on this call as well, which is Dr. Alan Beggs. So he's also available to help field questions. So thank you.
In addition to those action steps that Patty indicated on her slide, or Sarah read off of Patty's slide, the other action is to be more aware about the CMDIR and your involvement there. And if you have any questions, please reach out to Stacy or myself. Again, Sam Jacobson is a person in our community that's been willing to add some hours to her already full-time job in order to support moving the CMDIR forward.
Should we just confirm that participants can unmute and speak?
It's not set up that way, Alan. They're set up to do Q&A through
Q&A
button and/or chat. And there's nothing in there right now. Any updates you have for us would be great while we wait for a question to come in.
Yeah, I'd be happy to. I'd like to actually just focus on the natural history studies for a moment, which Gus and both Gus and Mark talked about. And just to emphasize how critical and important these are, because we really need to kind of understand what happens. As you know, mammalian myopathy is a very diverse condition. Some children are very weak right from birth, and others only develop weakness later or maybe weak throughout their lives. So the natural history study, an approach that Gus talked about, is what's called a prospective in-house clinical natural history study, and would involve you folks, members of the community, and your children coming in periodically to be seen by clinicians. And there's also now, we're having some discussions and I'm having thoughts about how to create what are called patient, PRO, or patient-reported outcomes. And this is essentially where you guys actively participate in helping to collect information by answering questionnaires, filling out information from home, perhaps doing some simple tests yourselves, answering questions about what you're able to do, standing, sitting, standing on one foot, not standing at all, how well you can swallow, things like that. And so I think that this is something to look forward to, that we hope to be developing some of these tools. Whenever you do a clinical study, you need to validate the tool. You need to kind of understand the relationship between what you're measuring and the actual patient experience, your ability to do things. So we hope in the coming year or so to start asking questions. I hope that all of you can try to come to the CMDIR conference this summer. And we'll ask you about the things that you can and can't do in your lives and what's important for you. And then from that, hopefully develop these questionnaires and tools that we would be able to measure and quantify, effectively try to put a number on your various abilities and disabilities, so this can be measured. So I'll leave it at that, but it's something that we're looking forward to engaging with all of you with. Mark, you're muted.
Yeah, Ellen, can I ask, are there resources right now for those that have not been confirmed with their exact gene variant to take those steps?
Yeah, I mean, actually, one of the critical aspects here is understanding the genetic basis for each person's condition. And as you saw, there are anywhere from 11 to 12, 13 genes that can cause Mammalian myopathy. And while there may be some uncertainty when we say those numbers, it's not a question of which genes cause a muscle condition. It's really what we decide to consider to be Mammalian myopathy. And as Gus showed you, one of the unifying features here is a defect in the muscle contractile apparatus and the thin filaments. And so that's one of our criteria. So the numbers are not exactly critical, but clearly there are differences between what the underlying cause of the weakness is, whether it's a mutation in the nebulin gene or a mutation in the KLHL41 gene or so on. And so that might lead to differences in how the condition progresses over a lifetime. It can also have major implications for which treatments might work. So a particular therapy, so you saw some of the therapies that AFBS is supporting research on. Some of them are gene replacement therapies. Those would only work for the particular gene that's defective in a given individual. So bottom line is, if you haven't already had a molecular diagnosis, that's an important thing to do. And how do you do that? Well, many physicians can now order clinical tests for their patients. And then if you get that result, that's something that you would want to let us know, let AFBS or the CMDIR if you register with them, hopefully. But if you haven't been able to do that, AFBS can help. My research lab can help, and other investigators in the field can help. So contacting Stacy Cassette, and when you register, you can be put in touch with resources to help potentially help cover the costs, or certainly there are many individuals who've enrolled in my own lab's research study with Casey Giannetti, who is traveling today in a train, and I think she had to drop off. But Casey, as a genetic counselor, can explain a lot of the resources that are available to you. And individuals who enroll with us, we also help to arrange to get genetic testing done. So the bottom line is there's many ways to achieve that. So please reach out and ask lots of questions.
Thanks, Alan. I think going back over the CMDIR quickly before we hit Christina's good question is that we do have about 250 people currently registered as Nemliners. And I think we have probably about 70 of those that are not confirmed. So still trying to figure out a path to get them full confirmation. In addition, we talked about doubling our numbers, so trying to find an additional 250 people that have not yet engaged in CMVIR registry would be great. So if you have or know people that would be interested, again, I think the best path is to contact Stacy probably until we can set up Sam with an email and registering address, or just go and start your own profile at cmdir.org. Stacy, do you want to put your best contact email in the chat as well? And then Gus, maybe you and I can field this question about, why if we have an FDA approved drug that seems to be working on zebrafish, can't we just start getting right into a clinical trial right away?
Yes, because still we still need the natural history study to decide how to measure the effect of that drug in the patient.
I think technically also the FDA would consider that an off-label use. It's not been cleared for the use in the diagnosis of Melanin myopathy. So any of these drugs that we find would be considered an off-label targeted use. And so certainly that's been done in the past. It's been done for, they've used epilepsy drugs for anxiety disorders and found success. And then later they've gotten approvals. So there potentially could be some targeted approaches that shortcut the system. But for now, safety has to be our very, very, very first decision that we're making. And so trying to take the steps and process that were so well articulated by that 5-step process of achieving, full admission of a medicine to treat the diagnosis are probably there. Okay, well, it's 10:58. We are available. If anyone has questions at any given time, please reach out to any of us. This, again, will be recorded and shared probably early next week. Anything else from the panelists? Any statements that they want to make before we sign off? Oh, Laura's got a question. Okay, hold on. For the treatment, candidate myostatin inhibitor, is it specific for NM patients? If so, what is the maximum extent of increasing muscle mass or strength? Is it sufficient for NM patients?
Well, yeah, this is the discussion, and this is why we are funding this grant to answer exactly those questions. At this point, the group should have some more results, because what I mentioned was the original proposal. So they will be giving a talk at SciFam meeting, and again, we will for sure bring them to future webinars. But yeah, I mean, the myostatin inhibitor has been tested in Duchenne with not very much luck. But now there's another clinical trial going on for SMA patients, and the results so far are promising. So yes, it's definitely a possibility. That's why we are studying that, but we don't have specific answers to your specific question.
As I understand it, and Alan and Gus can correct me if I'm wrong, but what myostatin might do is create more of the same muscle. So if you have a genetic error which is causing a weakness pattern, you'll create more weak muscle. You won't necessarily fix the development of the muscle structure, which is caused by the genetic problem inherently. So you would have a quantity of muscle that would be greater. And so if you're mildly weak, you might gain some strength from that action. Obviously, mitigating how much muscle is being added for these patients is critical, because you don't want to have an over addition of muscle. So yeah, the fine tuning these types of things is really important. And I again encourage all of you guys to attend in person if you can, preferentially, otherwise virtually at SciFam, when a lot of these researchers will be presenting.
Yeah, that's exactly right, Mark.
Okay, I'm going to give the one awkward pause before we sign off and see if there's any more questions. Okay, well, great. Thank you, everyone. We appreciate your attendance and participation and questions. Please keep them coming. It really does shape the future for those with mammalian myopathy. Bye now.
And please look for the post-webinar survey. The link is going to be sent to everybody along with the recording of this. Thanks, everyone.
Bye.
Bye-bye.